Good morning, everyone. Today we are going to have discussion on sense organ in fishes. Okay, so first in this class we are going to see what is sense organ. Okay, then what are the different type of sense organs present in the fishes, and then how they function. Okay, so we actually study these three things: what is sense organ, what are the different type of sense organs in fishes, and how they function. Okay, so in the today's class, first we will discuss about what is sense organ, then different type of sense organ which are present in the fishes, and then we will take the eye as a one sense organ, and remaining we will see in the next class. Okay, so for today class, up to this we will discuss. Okay, so first, sense organ in fishes. So sense organ are actually the specialized organs or a type of organs which are made up of different type of sensory cells. Okay, and these different type of sensory cells. So these organ actually receive the different type of stimuli from the external environment. Okay, the stimulus may be the physical or it may be a chemical stimulus. So basically, these sensory cells or the sense organ, they receive the physical and the chemical stimulus from the environment, and then what they do, they simply transmit it to the central nervous system, so that the body respond in such in a definite manner for any external stimulus. Okay, so the organ which receives the physical or the chemical stimuli from stimulus. Okay, means there is a different sense organ for smell, for light, for hearing, for taste, for touch, and for temperature. Okay, so they have different organs for different type of stimulus, or they have a different organ for different sensations. Okay, so here are the list of some of the important sense organs which are found in the fishes. So the number one is the sense organ for sight, or the photoreceptor, that is eye. Then the organ of hearing, that is internal ear or membranous labyrinth. Then the chemosensory organ, there are basically two type of chemosensory organs which are actually the sensory organ which sense the chemical stimuli. The one is the gustatory and another one is the olfactory. Gustatory sense organ is related to the taste and the olfactory is related to the smell. Okay, then the lateral line system is also a sense organ which sense the stimuli arise due to the touch. Okay, then the electroreceptor organ, there are basically two type of electroreceptor organs in the fishes. One is the ampullary organ and another one is the tuberous organ. Okay, so these are the list of sense organ which is found in the finfishes. So today we will take in detail the eye. Okay, okay, so as I have told, the eye is the organ of light reception. Okay, so or this is the organ which converts the light into the sight. Okay, so this is an organ, photoreceptor organ, and the structure of the eye resembles with the eye of the other vertebrates. Okay, so there is a functional as well as the structural resemblance of the eyes with the other vertebrates. There are some peculiar things about the fish eye, so that we will see here. In fishes, the eye does not have a lid or the covering, okay, that covers the eye. Like in the human and other vertebrates, what happen in general, most of the animals have the eye, which is actually have the lid or a protective covering over the eyes. But fishes does not have a that kind of lids, okay. But there is a one group of shark which is known as Galeorhinus. They have the lid like transparent nictitating membrane, okay. Otherwise fish eye doesn't have the lid or the lid like structure. And the another peculiar thing about the fish eye is the fishes have monoocular vision. The mono means single and ocular means eye. Mono is there's a Greek origin and ocular is the Latin origin. So monoocular okay, the humans have the biocular visions, okay, but fishes have the monoocular vision. So means two different eye altogether see two different objects, okay, because their position is lateral in the body, and because of this monoocular vision the field of view is considerably enlarged, okay. So fish can from one side fish can see from forward to the backward, okay, and from another side from forward to the backward. So this is about some of the special or the unique things about the fish eye. Now we will before discussing the physiology or how this organ function or how this organ converts the light into the sight, first we have to see the structure of the eye, okay. So we will see the structure. So the structure have basically four outermost transparent layer of the eye, okay. So this is the cornea, which is the outermost layer of the eye.
स्लाइड चेंज करिए मैम, स्लाइड वही है।
स्लाइड तो मैंने चेंज की है यहाँ पे। अच्छा
अब आया,
आ गया
वो कनेक्टिविटी इशू है थोड़ा, ओके। So this is the outermost layer, which is known as the cornea, the transparent layer, followed by the iris. Iris is region in the eye which help in the positioning of the lens, okay. Then this lens, and then this region is the eyeball, okay, which is followed by the pigment layer or the retina. And from here this eye is connected this is the diagrammatic representation of the teleused eye. This is the outermost, the transparent cornea, followed by the iris. Iris is actually the structure in the eye which help in the positioning of the lens, okay. And then this is the eyeball region, and the last one is the retina, or the region where the pigment cells which are actually responsible for the absorption of the light and converting it into the sight signal. And these sight signals are going to the brain through this optic nerve, okay. So we will see each part one by one. So the first is the cornea. As I have told, this is the most outer or the anterior most part of the eye, and it is composed of three layers. The outermost layer is known as the corneal epithelium, followed by the middle layer, which is known as a corneal stroma, and the innermost layer of the cornea, which is known as the corneal endothelium. Okay, so this tri-layer of the cornea, it doesn't have any pigments. That's why it has the transparent appearance. And this cornea, it helps in the retention of the water and it prevents the drying of the eye. Okay, then this iris, which is just lie behind the cornea. Okay, so iris is behind the cornea and in front of the lens. Okay, and it is composed of the pupil, which actually controls the amount of light that reaches to the retina. Okay, so this pupil diameter, okay, that is controlled by the reach to the retina. Okay, so in this, iris actually helps in controlling the amount of light, okay, to reach the retina. Okay, then third one is the lenses. Okay, the lens is a transparent ball like and it is composed of the non-collagenous protein. This lens is covered by the lens capsule and it is filled with the substances, a different type of matrix substances, that is known as lens substances. Okay, now the accommodation of light. What happened, that is, there is some difference between the fish and the higher vertebrate eyes. And what happened in case of humans, what happened when the light falls on the eyes. Okay, when the light falls on the eyes, what happened, the pupil of the eye, it contracts or dilate. Okay, the contraction and dilation actually adjusts the amount of the light which is reaching to the retina. Okay, what happened, this basically there is a contraction or dilation of the pupil. But in fishes, what happened, so this contraction or dilation actually cause the contraction or dilation of the lens itself, na? But in case of fishes, what happened, because of the presence of one muscle which is known or lentis muscle, or the which is also known as campelluna of Hallery. Okay, this muscle actually help in positioning the lens inside the eye. Means how much light will reach to the retina, it is basically controlled by the positioning of the lenses. Okay, means the lens position actually control the amount of light that will reach the retina. But there is like in elasmobranches, okay, like sharks and rays, in which what happened, in elasmobranches, the light control mechanism is like the higher vertebrates or the human, where the pupil diameter is adjusted to um, control the amount of light. In teleos fishes, what happened, the position of the lens alters. Okay, the position of the lens actually control the amount of light. But in elasmobranches, the pupil diameter, okay, there is an adjustment in the pupil diameter, and that adjustment in the pupil diameter is because of the muscular iris. Okay, so because of this, the amount of light is controlled. Okay, otherwise in the teleos, there is basically the position of the lens, not the shape. Okay, now the eyeball. Okay, so the fourth part of the eye, that is the eyeball, it have three layers. Okay, the outermost is known as the sclerotic capsule, the middle one is known as choroid layer, and the inner one is known as the retina. Okay, so in the eyeball, the three layers, the outer one is the sclerotic capsule, middle one is the choroid layer, and the inner one is the retina. So first we will talk about the scleroid sclerotic capsule. It is the outermost fibrous layer which is strengthened by the presence of the cartilaginous elements. Okay. Then the choroid layer, it is the middle highly vascular layer which have a dense network of blood capillaries, which is also known as rete mirabile or choroid rete. Okay. So this rete mirabile or the choroid rete is actually the organ which supply the extra amount of oxygen to the retina, because in the eye there is an excess requirement of the oxygen for converting the vision or converting the light into the sight. Okay. So if there is a normal diffusion, what happened? Basically the oxygen diffuse from higher concentration to the lower concentration. So what will happen from arteries to the veins, what will happen when the oxygen diffuse? What will happen? Only 50% of the oxygen will diffuse. Okay. And when there is the same concentration in the arterial and the venous blood, there will be no diffusion or exchange of the oxygen. Okay. So this rete mirabile have different structure that supports the pass of the oxygen even against the functional physiology of the eye, how this rete mirabile or the choroid rete help in providing the excess supply of the oxygen to the retina. Okay. Now the innermost layer is retina, which have three main structures or three main layer. One is the light sensitive layer of the light sensitive cells, visual cells, basically the rod cells and cone cells. Then there is a layer of the bipolar cell. Actually bipolar cell connected to the rod and cone cells, or you can say that the bipolar cells are connected to the visual cells, and to the one end, and to the another end it is connected to the ganglionic cells. Okay. So means at the top there is a light sensitive cell, then the bipolar cell, and then the ganglionic cells. So this bipolar is connecting with, at one end it is connected with the visual cell, and at another end it is connected with the ganglionic cell. And this ganglionic cell is further connected to the optic nerve that sends the signals from eyes to the brain. Okay. Now as I have told, the retina have the light sensitive visual cells, cone cells and rod cells. So these retina have two type of light sensitive pigment or photopigment. One is known as the rhodopsin and another one is the porphyropsin. So here we will see the detail about the photopigments in fishes.
मैम
Yes.
मैम ये गैंग्लियोनिक सेल जो है और बाइपोलर सेल्स, ये भी रॉड या फिर विजुअल सेल्स को और गैंग्लियोनिक सेल को कनेक्ट करता है।
हाँ।
और गैंग्लियोनिक सेल भी बाइपोलर सेल्स और ऑप्टिक यानी कि विजुअल सेल्स को कनेक्ट करता है। तो दोनों का कनेक्शन जो हो गया वो तो सेम हो गया मैम।
कहने का मतलब है जस्ट सी दिस इज अ सैंडविच लाइक सिचुएशन। टॉप है, बॉटम है, दो स्लाइसेस हैं सैंडविच की। ठीक है। एक टॉप स्लाइस है, एक बॉटम स्लाइस है, और बीच में आपकी फिलिंग है। कोई भी फिलिंग हो सकती है, चीज की फिलिंग हो सकती है, वेजिटेबल्स की फिलिंग हो सकती है। तो अगर आप सैंडविच को एज देख रहे हैं, जो तो टॉप वाली जो लेयर है, जो ब्रेड की टॉप लेयर है, दैट इज द विजुअल सेल्स।
रॉड एंड कोन। हाँ।
रॉड, मे बी रॉड सेल्स और दी कोन सेल्स और बोथ द सेल्स। ओके। देन जो आपकी फिलिंग है, जो चीज या जो सैंडविच की फिलिंग है, चीज, वेजीज, आलू टिक्की, व्हाटएवर यू प्रेफर,
बाइपोलर
सेल। ठीक है। और जो नीचे वाली जो लेयर है, दैट इज द गैंग्लियोनिक सेल। इतना क्लियर हुआ? तो ये जो सेल जो है वो अगर बीच में है, अगर वो फिलिंग की साइड में है, तो उसका कनेक्शन किससे है? ऊपर कोन और रॉड सेल्स के साथ है, और नीचे आप
गैंग्लियोनिक
सेल्स के साथ है। अब ये जो गैंग्लियोनिक सेल्स हैं, जो कि बाइपोलर सेल से कनेक्टेड है, ये गैंग्लियोनिक सेल्स का कनेक्शन है ऑप्टिक नर्व के साथ। ठीक है। और ये जो ऑप्टिक नर्व है, ये आपकी कनेक्टेड है सेंट्रल नर्वस सिस्टम के साथ और
तो
ब्रेन।
यहाँ ऑप्टिक नर्व का मतलब रॉड एंड कोन आएगा या फिर कुछ और आएगा?
समझी नहीं? आई एम नॉट।
जो ऑप्टिक नर्व का फंक्शन होता है रॉड और कोन से कनेक्टेड होता है मैम?
नहीं, ऑप्टिक नर्व जो है रेटिना से कनेक्टेड है। ठीक है। और रेटिना की 3 लेयर्स हैं। सबसे पहली लेयर जो है वो विजुअल सेल्स की लेयर है, फिर बाइपोलर सेल की लेयर है, उसके बाद गैंग्लियोनिक सेल की लेयर है। आई बात समझ में?
ओके मैम।
अभी हम नेक्स्ट स्लाइड में इसका स्ट्रक्चर देखेंगे, देन इट विल बी क्लियर टू यू। ओके
ओके मैम। ओके।
So now we will first talk about the photopigments, okay, or the photoreceptor pigments which are present in the eye. Okay, so basically there is a 2 type of photopigments. Okay, so first is so if I'm talking about the visual photopigment, it have 2 parts. One is the retinal, which is a vitamin A derivative, and another one is the opsin, which is a glycoprotein. Okay, so this retinal and opsin together forms the photopigment. So there is basically 2 category or 2 broad group of the photopigments. One is the rhodopsin and another one is the porphyropsin. So rhodopsin is purple color photopigment and porphyropsin is the rose color photopigment. So this purple color rhodopsin is actually have opsin and aldehyde derivative of vitamin A. Okay, so the aldehyde of vitamin A plus opsin, it forms the rhodopsin, which is responsible for the monochrome vision, okay, black and white vision you can say. Okay, now the porphyropsin or the rose color pigment, which is made up of opsin and 3-hydroretinal. Okay, this is also a derivative of the vitamin A. So this opsin plus 3-hydroretinal, this which forms the rose color porphyropsin, is responsible for the color vision. Okay, so what happened, this cone, I have told there is a 2 type of visual cells. One is the cone cells and another one is the rod cell. So what happened in the cone cells, there are different type of photopigments. So different type of photopigments means it have the photopigments which have the same opsin, but the retinal or the vitamin A derivative is slightly different, okay, in structure. So what happened, because of this slight difference, the photopigments which is present in the cone cell is able to absorb a different range of light wavelengths. Means it is able to absorb the wavelength which are of different colors. So what happened, so there is actually the mixed stimulation of the different cone cells, maybe the red cells, green or the blue, and because of their optimal absorbed wavelength, it provides the perception of varied color. That is why the pigment which is present in the cone cells, porphyropsin mainly, is responsible for the color vision, and in the rod cell it have only one kind of pigment which is known as the rhodopsin, which is made up of opsin and aldehyde derivative of vitamin A, is responsible for a single type of monochrome vision. Now we will see the structure of the rod and cone cells. So the basic difference between the rod and cone cell is the length of outer segment. The number of discs present in the outer segment is more in the rod cell as compared to the cone cells. Okay, and even the synaptical terminal is also broader in the cone cell as compared to rod cell. So this presence of this disc in the outer segment is the major structural difference between the rod and cone cell. And another difference is the presence of different type of photopigments. Rod cells have rhodopsin, cone cells have porphyropsin. Rod cell rhodopsin is purple in color and cone cell cone cells pigment is rose color in red, rose red in color. Okay, so now here we'll see the difference between the rods and the cones. Okay, so the very first difference, as I have told, the difference in the presence of the photopigments. The rods have the rhodopsin and the cones have the porphyropsin. Then the second difference, it is the rod cell are highly sensitive to the light as compared to the cone cells. So the sensitivity is high in rod cell, that's why they are nocturnal for night vision. Okay, so rod cells, basically the cells or the pigments which is present in the rod cell is responsible for the vision in dark or night. Okay, and this cone cell lower sensitivity to light, that's why it is diurnal or it is used in the daytime vision.
Ma'am, मैम
Yes.
लोअर सेंसिटिविटी कैसे हो गया? कौन मैम, कौन तो सब वेवलेंथ को देख रहा है, मैक्सिमम वेवलेंथ को ऑब्जर्व कर सकता है,
लो
सब कलर को?
हाँ हाँ मैं बता रही, यहाँ पे लो सेंसिटिविटी से
इस टर्म से आप बोल रहे हैं, मतलब कि नंबर ऑफ
हाँ,
डिस्क कम है इसलिए?
नहीं नहीं, नंबर ऑफ सेल्स, हाँ लो सेंसिटिविटी का मतलब है कि नंबर ऑफ फोटो पिगमेंट्स कम है कोन सेल्स में, कहने का मत। और दूसरा, नंबर ऑफ फोटो पिगमेंट्स कम हो गए तो क्या होगा, जो लाइट को डिटेक्ट करने की जो सेंसिटिविटी है वो रॉड सेल में ज्यादा है, कोन सेल में कम है। क्योंकि अगर लाइट को डिटेक्ट करने की सेंसिटिविटी कोन सेल में ज्यादा होती तो कोन सेल, रात में क्या होता है, लाइट कम होती है ना अंधेरे में, नाइट या डार्कनेस में लाइट बहुत कम है, ठीक है। लेकिन रॉड सेल्स जो है डार्कनेस में भी विजन को हेल्प कर रही हैं। क्यों? क्योंकि उसमें नंबर ऑफ फोटो पिगमेंट ज्यादा है, उनकी लाइट के लिए सेंसिटिविटी ज्यादा है। कोन सेल्स जो है ज्यादा, उसमें अलग अलग टाइप के फोटो रिसेप्टर हैं, इसलिए कलर विजन है, लेकिन उनकी सेंसिटिविटी कम है। मींस लाइट को लेके उनकी सेंसिटिविटी कम है। अगर लाइट डिम है या डार्कनेस है, सो यू विल नॉट
वो
बी,
डिफरेंशिएट नहीं कर पाएगा।
हाँ ठीक है। सेंसिटिविटी का मतलब है लाइट को डिटेक्ट करने की सेंसिटिविटी।
ओके मैम।
Is clear? Okay. So
यस
now difference in another difference, the rod cells have more photopigment, so they have the greater photon capture capacity and the high amplification, whereas the cone cells have less photopigment and the less amplification. Then the pigments in the rod cells, they get saturated even at very low light or low level of brightness, okay. And in the cone cells, saturations occurs only in the intense or the bright light. The resolution is low, resolution is high, okay. In cone cell high resolution, in rod cell low resolution. Then the slow response kinetics and low integration time means जब ये जो rod cells हैं, जब इन पे lights पड़ेगी तो इनका जो response का time है वो slow है, लेकिन cone cells का light पड़ने के बाद जो response time है वो ज्यादा है, ठीक है। Then जो आपकी रॉड सेल्स हैं, दे आर सेंसिटिव टू द स्कैटर्ड लाइट, ओके। अगर स्कैटर्ड लाइट है उसके लिए वो ज्यादा सेंसिटिव है, और कोन सेल्स जो है वो जो डायरेक्ट रेज आती हैं टारगेट से निकल के उसके लिए सेंसिटिव है। देन इसमें से वन टाइप का फोटो रिसेप्टर होता है रॉड सेल्स में, दैट्स व्हाई इट इज रिस्पॉन्सिबल फॉर मोनोक्रोमैटिक विजन और द ब्लैक एंड व्हाइट विजन, ओके। और इसमें आपका जो कोन सेल्स है, इसमें सेवरल टाइप ऑफ फोटोरिसेप्टर्स हैं विद डिफरेंशियल सेंसिटिविटी, दैट्स व्हाई द कोन सेल्स हैव अ कैपेसिटी फॉर द color vision. So that is the main difference between the rod and cones. Everyone?
ह्यूमन में भी सेम केस है, सब चीज
Yes.
सिमिलर है?
Yes. Okay. Now, as I have told, the eye is an organ, okay, a photoreceptor organ which converts the light into the sight. Means a light falls on the eye, okay. The light which is coming from one object is falls on the light on the eye, okay. Animal's eye, here we are talking about fish, okay. So the light which is emanating from the object is falls on the eye of the fish. Then how the fish converts this light into the sight or this light into the vision? Means agar suppose a fish is seeing a rock on the way, when it is swimming there is a rock in a bottom. So suppose if the light which is emanating from the rock to the fish eye is simply a light, okay, a wavelength which travels to the retina. Now how this retina converts this light or this wavelength to the vision, okay, or how the nervous system or the brain it interprets, okay. So here we will see. So what happened when this photopigment which is present on the retina, it absorbs the light, what will happen? The opsin is get detached from the photopigment, whether it is rhodopsin or porphyropsin. What will happen? The retinal will get detached from the opsin. So this freed opsin, so what happened when this freed opsin is formed or the opsin is get separated, the product is colorless. That's why this process is also known as bleaching, okay. So what happened? This free opsin, now it is chemically active. So because it is chemically active, it initiates the series of chemical reaction. Okay, it initiates a series of chemical reaction. So it actually, this freed opsin stimulate one GTP linked protein, which is known as the transducin. Okay, now this transducin activate one enzyme which is known as cyclic GMP phosphodiesterase. So when this cyclic GMP phosphodiesterase gets activated, it catalyzes the breakdown of cyclic GMP into the non-cyclic form. Okay, so it basically converts the cyclic GMP into the non-cyclic form. Here we will see. So when this cyclic AMP is converted into the non-cyclic AMP, what will happen? The concentration of the cyclic GMP is now lowered in the cell, inside the photopigment cell. So when there is a lowering of the concentration of cyclic GMP, what will happen? It will triggers the closing of sodium channels. So what will happen? When cyclic GMP is sufficient inside the cell, the sodium channels are open. So sodium ions are constantly coming inside the cell. But when the concentration of the cyclic GMP is lowered, what will happen? This sodium ion or the sodium channel get closed. So what happened now? There will be no transport of the sodium, transmembrane transport of the sodium ion. So what happened? Because sodium ions are not coming inside the cell. So what happened? There is an increase in the positive charge on the plasma, on other side of the plasma membrane. So it causes the hyperpolarization of the plasma membrane or increase of the charge on one side of the plasma membrane. So because of this hyperpolarization, what will happen? The normal secretion of the neurotransmitter gets stopped. So what happened? This hyperpolarization is triggers the graded potential inside the bipolar cell. Okay, now this bipolar cell, because there is a graded potential in the bipolar cell. So now this change in the membrane potential basically sends the signals to the brain. Okay, and then brain converts this light into the sight or a image of a object. Here we'll see in the diagram, it will be more clear to you people. Just see. This is your photopigment cell. This is the disc, okay, which is present in the outer layer of the photopigment cell. In the disc, there is a visual pigment. See, here is a rhodopsin. So what happened? When light falls on the rhodopsin, what will happen? Opsin get detached from the rhodopsin. So now this opsin activate this G protein, okay, or the transducin. The activation of this transducin cause the activation of the another membrane linked enzyme which is known as cyclic GMP phosphodiesterase. Now this cyclic GMP phosphodiesterase triggers the conversion of cyclic GMP into the non-cyclic GMP. So when there is a, because this cyclic GMP is now converting into the non-cyclic GMP, now there is a low concentration of the cyclic GMP inside the cell. So because of this lowering of the cyclic GMP, this sodium ion channel get closed. Now there is no sodium ion coming inside the cell. So what happened? There will be the increased positive charge on this. So it caused the hyperpolarization of the plasma membrane. And this hyperpolarization actually caused the graded potential in the bipolar cell. Now this bipolar cell sends the signals to the ganglionic cells. And now these ganglionic cells send the signals to the optic nerve. And optic nerve actually sends the signals to the brain. And the brain, this signal or the graded potential actually converts the light into the image or sight. Okay, so this is the basic actually phototransduction physiology or mechanism of the eye, how the eye actually functions in the formation of the vision, formation of the image or in vision. Okay, so now the light sensitivity of the retina. Okay, what happened? The retina also contain one layer of melanin, okay, and choroid coat. So because as I have told, both the rod and cone cells, they both are the very sensitive to the light. So what happened? This when there is a bright light, this melanin, here we can see, okay, when there is a when there is a bright light or a daytime, what happened, this melanin sheath increases size and it covers the rod cells, okay, because rod cells are very sensitive to the bright light and they easily get bleached, okay. And in the dark, when light is less, what happened, this melanin actually migrated towards the end, and this now rod cell is nearer to the lens, okay. So this is the adaptation of the rod and the cone cells or the visual cells in dark or light. Now we'll see the mechanism how the fish eye adapt, because when fish swims, or there is both the period in the day, the night and the day, then how the fish eye adapt to the light and dark, okay, adaptation. So what happened, when there is a bright light, a very light sensitive rhodopsin in rod cannot be regenerated. What happened, when light falls on the rhodopsin, opsin get dissociated from the rhodopsin. So when it again have to absorb the light, opsin needs to be get attached to the retinal to make the rhodopsin. So this process is very slow, okay. The regeneration of the broken rhodopsin is slow. So what happened, because in the bright light, so your rhodopsin remains inactive, okay. So what happened, as a result, because your one of the light sensitive pigment is inactive, so the less light sensitive pigment which is present in the cone cells, that are porphyropsin, they become active in the normal or the bright light, okay. What happened now, when the fish move from the bright condition to the dark, what happened, the photopigments of the cones, they are not too sensitive to detect the light. So what will happen, it will basically the rhodopsin vision is returned, okay, and this because of the regeneration, and this process is called dark adaptation, okay. Now what happened, again fish move from dark to the bright condition, then the very light sensitive rods are suddenly overwhelmed and it produce a glare kind of sensation, okay. And then again the less sensitive cone cells, they resume the activity. So this is why, means when there is a dark, then there is a rhodopsin takes the function, okay. And when there is a light, the porphyropsin or the cone cells resume the activity. So because of the rod and cone cell activity, the fish eye adapt to the darkness and light, okay. So now, as I have told, the retina required more oxygen for proper functioning, okay, because the retina is a avascularized organ, okay, और उसको बहुत ज़्यादा ऑक्सीजन चाहिए. So क्या होता है, if you remember our respiration class, in that respiration class we also studied one effect which is known as a root effect, okay. This root effect occurs in the fish hemoglobin. What will happen if there is a decrease in the pH? This red line shows the root effect, okay. If there is a decrease in the pH, means if the pH is acidic, what happened, if the pH is acidic, this graph shifts right as well as downwards. This line shifted right as well as downwards. Why? Because if the pH is acidic or low, even if you increase the oxygen concentration, the hemoglobin cannot be saturated with the oxygen. Means if pH is low, the hemoglobin get disassociated from the oxygen get disassociated from the hemoglobin. So basically root effect shows the loss of cooperativity at low pH. Means if pH is low, even if you increase the oxygen concentration or the pressure of the oxygen, the saturation of the hemoglobin with oxygen does not occur. Okay, means the hemoglobin cannot be fully oxygenated even at high oxygen pressure when pH is low. Okay, and because of this effect, because of this root effect, the choroid rete, which is basically a network of the blood vessels in the fish eye. Okay, it unload the more oxygen in the retina against a high concentration gradient. Okay, this we have also studied in the swim bladder, if you remember the last semester class. So what happened? The fish, actually the blood in the fish is received from the afferent arteries of the pseudobranch. Okay, and this pseudobranch actually received the oxygenated blood from the first gill. So what happened when the blood is reached to the pseudobranch? It is titrated at the pH value, which is sufficient to induce the root effect. Means it titrate the blood to the lower pH. So what happened when the blood reaches to the choroid? Okay, what happened? A very small amount of acid is get released from the retina. So because the acid is released, what it causes? It causes the reduction of the pH. And because of the reduction of the pH, the hemoglobin which is present in the choriocapillaries, they started showing root effect. And because of this root effect, what happened? Even the oxygen tension is high, but because pH is low, the oxygen get liberated from the hemoglobin. So because of this, even this oxygen, okay, is diffused back to the choroid rete mirabile, and because of this, it generates the high oxygen partial pressure, which is required to ensure the sufficient oxygen supply to the retina. Okay, so this is how the root effect affects, okay, or responsible for diffusion of more and more oxygen to the retina through the choroid rete or the rete mirabile. Okay, so this is how the oxygen supplies goes to the